morning, students of class 11. Welcome to today's recorded class of biology. We will continue studying about the cell from the chapter Cell, the Unit of Life. Before starting with today's topic, we will revise in short what we have learned in the last class. So in the last class, we studied about endoplasmic reticulum, that its structure is a network of tiny tubules. Endoplasmic reticulum are of two types: rough endoplasmic reticulum synthesizing proteins and smooth endoplasmic reticulum synthesizing lipids. This endoplasmic reticulum, it is in continuous with the Golgi apparatus. This Golgi apparatus receives the proteins and the lipids sent by the endoplasmic reticulum. The Golgi apparatus has two phases, is it not? The cis phase and the trans phase. The cis phase is a forming phase and the trans phase is a maturing phase. After this, we came to know the functions of the Golgi apparatus. The Golgi apparatus modifies the proteins and the lipids which it gets from the endoplasmic reticulum and also packages them in form of small vesicles. So after this, what we are going to learn in today's class from the chapter. Our learning outcomes for today's class are to describe the structure of lysosomes, vacuoles and mitochondria. We are going to name the different parts of the cell organelle mitochondria. We are also going to list the functions of mitochondria. Fine. So let us get started with the topics. So we were studying about the endomembrane system and we have studied the two organelles of endomembrane system already, endoplasmic reticulum and Golgi apparatus. The next organelle which is a member of the endomembrane system is the lysosomes. What are lysosomes? Lysosomes are also some membrane-bound sacs or vesicles. These are present in the cytoplasm. These lysosomes, they contain the proteins and the lipids which comes from the Golgi apparatus. This lysosome basically consists of the enzymes, the breaking enzyme, the lysine enzymes, which will break down carbohydrates, proteins, lipids and nucleic acids. Some of these enzymes which are present in the lysosomes are the lipases which breaks down the lipid, the proteases which break down the protein, the carbohydrases which break down or digest the carbohydrates. And these enzymes of lysozymes, they work better in a low pH, that means in an acidic medium. The content inside the lysosome are kept in always acidic medium. Another organelle of the endomembrane system is the vacuoles. Every cell has a vacuole, small or large. Vacuole is also a membrane-bound space found in the cytoplasm. We know that the plant cells, they have got a large vacuole in comparison to the animals. These vacuoles, they store water, the cell sap in plants, maybe some excretory products and other materials which the cell is not requiring much. This vacuole in plants, it occupies 90% of the site of the area of the plant cell. And the vacuole in plant cell is so large that it is centrally located. Generally, the vacuole of the plant cell is called as the central vacuole. And this vacuole shifts the nucleus and other cell organelle towards the periphery. The membrane, the plasma membrane of this central vacuole present in plants is very special. We call it tonoplast. Why it is very special? Let us see. It is very special because the tonoplast facilitates or helps in transport of ions and materials against the concentration gradient. Remember, we studied active and the passive transport. That means tonoplast is responsible or is functioning active transport to pump the ions and some other materials which are already there inside the vacuoles. Still, this membrane is pumping those ions inside the vacuole. So, tonoplast has large number of proteins which acts as pump to pump the ions and these materials inside. Apart from the plant cells, vacuoles are also present in animal and protists than the plants. Take the example of paramecium. The paramecium and amoeba, they engulf the food which is present in their surrounding and they form a vacuole. This vacuole is very small and this vacuole consists of the food particle. This vacuole is called as a food vacuole. In case of amoeba, after consumption, after utilizing the food, the excreta which is produced, it is stored in another form of vacuole which is called as the contractile vacuole. And this materials inside the contractile vacuole is secreted out by the amoeba. You see in this diagram how the stored materials, the excretory materials are released out by the amoeba through the contractile vacuole, right? So by this time, what we have studied, let us see, we were studying actually the endomembrane system. In eukaryotic cell, we started studying the plasma membrane, then we studied cell wall, then we started endomembrane system. In the last class, we studied about the endoplasmic reticulum and Golgi complex of endomembrane system, and today we started studying the two more components of endomembrane system, lysosomes and vacuoles. That means we have completed the endomembrane system, their functions, how they work and how is the interconnection of their functions. Now we will take up another cell organelle, that is mitochondria. You should be alert that mitochondria is not a part of the endomembrane system. Mitochondria is another organelle present in the cytoplasm. A very important characteristic of the mitochondria is that mitochondria has two membranes. These mitochondria are very clearly visible under microscope when stained, the most clear cell organelle. The number of mitochondria, how many are there in the cytoplasm of a cell, the shape, okay, then the size, all these things are varied. In different cells, different types of mitochondria, different shapes of mitochondria will be present. Take the example of a muscle cell. Muscle cell, needs so much of energy to do work. So in muscle cell, the mitochondria, number of mitochondria will be very high. So based on the type of cell, the number, the size, the shape of the mitochondria varies. Generally, the mitochondria are sausage or cylindrical shape. This mitochondria is double membrane, as I already told you. Now you can see in this image that this is the outer membrane and the outer membrane is a smooth membrane. But can you see the inner membrane, which is not smooth but it is folded by forming some infoldings, not outfoldings, infoldings. The membrane is not smooth. Membrane is forming foldings. So this inner membrane divides the lumen, the space inside the mitochondria, into two compartments. Very important point. Two compartments. What are the two compartments? The intermembrane space between the outer membrane and the inner mitochondrial membrane, and the matrix inside the inner membrane. These infoldings which this mitochondria is producing are called as crista, or in plural cristae, towards the matrix. The function of this crista, for formation of crista, is to increase the surface area. More is the surface area of inner membrane, more efficiently the mitochondria will be able to do the function. Both the membranes of mitochondria, inner and outer, has specialized proteins. We know that plasma membrane is made up of proteins and lipids, but in the membrane of mitochondria, the protein is of a special kind. These proteins are the enzymes. In the matrix, you see, of the mitochondria, there are DNA, right? But the DNA is special. Why? The DNA is a circular DNA. It's like a circular DNA. You remember we studied plasmid? So mitochondrial DNA is like plasmid and it is circular. Apart from the circular DNA, the mitochondria has also ribosome. You can see these green bodies are ribosome. These ribosomes are also different. Why? Because these ribosomes are the prokaryotic ribosome of 70S. This matrix will also have RNA. The DNA, RNA and the ribosome, 70S ribosome, are responsible for production of protein inside the matrix or in the matrix of mitochondria. That means mitochondria is self-sufficient. And did you realize that mitochondria has all the components like a prokaryotic cell, like the circular DNA, like the 70S ribosomes? Do you know that mitochondria is actually a prokaryotic cell which have been living symbiotically in the eukaryotic cell from ages? In the course of evolution, the mitochondria has established the living inside the eukaryotic cells. This mitochondria, like a prokaryotic cell, also divides by the process of fission. Now, what are the functions of mitochondria? Mitochondria is the site for aerobic respiration. During aerobic respiration, energy is produced in the form of ATP. This ATP are produced because of this F1 and F0 particle which you can see in the image, which is present in the inner membrane of mitochondria. So F0, F1 particle is a kind of a special protein. More is the enfolding in the inner membrane, more is the F0, F1 particle, and more is the F0, F1 particle, more will be the ATP produced. So that's why mitochondria is called the powerhouse of the cell, which is synthesizing ATP, the currency of energy. Fine. So what we have studied in today's class, let us revise in short. We studied about lysosomes. Lysosomes are some membrane-bound vesicles which contains lysosomal enzymes, the digesting enzymes. Then we studied another component of endomembrane system, the vacuoles. Vacuoles are also membrane-bound sacs present in the cytoplasm. These vacuoles in plants are of very large size. The membrane of the plant vacuole has tonoplast. Tonoplast is important because it carries out active transport. Vacuoles are also present in the cells like amoeba and paramecium in the form of contractile and food vacuole. After this, we studied the structure of mitochondria. We studied that the mitochondrial structure is varied based on the type of cell it is present in. This mitochondria has two membranes, inner membrane and outer membrane. Outer membrane is smooth, whereas inner membrane has infoldings. The inner membrane divides the lumen of the mitochondria into two chambers. The outer is the intermembrane space between the two membranes, and inside is the matrix. Matrix is like the cytoplasm of a cell. Matrix has circular DNA like a prokaryotic cell. Matrix has 70S ribosome like a prokaryotic cell. And the matrix also have RNA like a prokaryotic cell. These infoldings which are present in the inner membrane are called as crista. These crista are special because this crista has F0, F1 particles. And F0, F1 particle are the enzymes made up of proteins which are involved in the formation of ATP. And with the help of this F0, F1 particle, mitochondria is able to produce large number of ATP in aerobic respiration. So, we have been calling mitochondria the powerhouse of the cell. You understood why? So, thank you for attending the class and happy learning. please watch the video again and again. If you don't understand something, please ask me.